Miss,
Yes.
for Edexcel and iGCSE, Cambridge and O level, the syllabus is same.
Approximately the topics are same, right?
Oh,
Topics, the concepts are approximately the same. There are just few extensions, and you will be entertained at the end for that, right? Don't get worried. Okay, so yesterday we were doing this bacterial cell, right? Okay, so we did studied that this is a bacterial cell. It was made up of peptidoglycan. We didn't mention or discuss the plasmids yesterday, right? These are the small circular DNAs, right? These are the plasmids. Children, are you listening? I couldn't hear any one of you.
Yes, miss.
Keep on
Yes, ma'am.
saying yes, no, or asking questions so that I would know that you people are here. Because
Miss, your
once
voice
I,
is coming very slow.
yes,
Your voice is coming very slow.
is that for all of you? All of
Miss,
you
I can
cannot.
hear others except you.
Okay, tell me others as well. That is my voice low.
No, ma'am. Fine for me.
Okay, then Nadeem, you have to do something with your speaker because I'm talking.
Okay, I'll try.
You'll try to do it that at your end. Okay. Now we have to move towards plasmids. Now plasmids are the small circular structures. These are only present in bacterial cells, right? These are small circular DNAs, and these are except the circular chromosomes. So you can say that they are made up of DNA because some of the genes of DNA might be on plasmids as well. You got it?
Can you repeat it?
Okay, I told you that yesterday we were talking about the bacterial cells. We studied few of the things, right? Then today we are going to study about the plasmids. Now, plasmids are the small circular structures like this. So these plasmids are basically made up of they are also made up of DNA, right? And these plasmids are very important for genetic modifications because it is easy to insert genetic material into them, right? What you can do, you can cut a specific portion of this plasmid, you can add the gene of interest into this plasmid, so as the bacterial cell will divide, these plasmids will also divide, right? And you can have a lot of your gene of interest. Okay, then we haven't so there are some ribosomes also present in the bacterial cell, right? They are floating freely in the cytoplasm. And then Then we talked about that we have a circular DNA as well. So there's a single chromosome as well apart from plasmid, right? That is made up of circular strand of DNA. You got it?
Yes, miss.
Okay, now we know that these bacterial cells could be in different shapes. So the shapes of bacterial cell could be they could be either spherical or they could be rod shaped or they could be spiral. Okay. And then there's one more thing about the plasmid, that is, okay, we know that there are some other genes that are present on plasmids, right? The genes that are on plasmids, there is an important function of them, that is, they carry genetic information to help the processes of the survival and reproduction of the bacterium. You got it? And for ribosomes, we know that they are involved in protein synthesis, and I think we have discussed the cell wall as well. Now we have to move towards another topic that is called as the specialization of the cells. Right? Now, what are the characteristics of a specialized cell? For a specialized cell, you know that they do one special job, right? Other than that,
Yes.
you can say they develop a distinct shape, they have special kinds of chemical changes that take place in their cytoplasm, right? If a cell is specialized, that means its structure is specialized to perform a specific function, right? A specialized cell has a special structure that will be supporting a special function. So for a specialized cell, it is responsible to do one special job. Apart from that, it has a distinct shape, right? Its cytoplasm is going to have special changes or special chemical reactions in their cytoplasm. Right? Now we know that the changes in the shape and the chemical reactions enable the cell to carry out its special function. If you're going to see the example of the examples of specialized cells could be the root hair cells and red blood cells. There are other examples as well. Are you getting it, class?
Yes.
Yes.
Yes, ma'am.
Okay. Now, if we see the red blood cells, how they are specialized to do their specific function, so we can observe that they look like this. They are quite, they look like they have a concave inside.
They have a biconcave shape.
They have a bioconcave shape. And they are packed with hemoglobin. And apart from that,
Ma'am,
yes,
ma'am, do red blood cells they don't have a nucleus, right?
yes, because they are made just to carry oxygen throughout their life. So there's no other, like they don't have to do other tasks. So that's why nucleus is not needed in them. So this hemoglobin is known as the red pigment. And obviously they contain no nucleus as well. And we have mentioned that they have they have the bioconcave shape. So if we talk about the root hair cells, so they look like this. This is how their structure looks like. And there you have a nucleus in them. Then this outer one is the cell wall. The inner one is the cell membrane. And here this inner one is the large vacuole. Actually, this large vacuole has a third layer. Let me draw that like this. Right? This middle one is the vacuole, and then you have a nucleus over here. Now, this look like this too. This root hair cell is to absorb water and minerals. Now you know that it has this finger-like projection. That is actually the root hair cell, but we can say that it is a finger-like projection, and due to that, it facilitates you. It's going to increase the surface area. Why do we need increased surface area? We need that for absorption. Absorption of what? Water and minerals, right?
Mineral ions.
Good. And we know that it has a large vacuole that is important for this large vacuole is important for to store more water, right? Then we can move towards the palisade mesophyll cells. Now, how do they look like? They look like this. And this is the second one, right? It looks like something like that. Okay, so in the here we know they have the outer one is the cell wall, right? The inner one is the cell membrane. Then we have this the in the middle we have this empty portion is mostly the vacuole, right? And here at these corners you have the chloroplasts like this, right? Now where are these palisade mesophyll cells present? Yes, anyone could tell me? Children, are you listening me? Yes, Miss Sam, are you here? Or you people can listen
Ma'am,
me?
under, yes, yes, under the upper epidermis layer.
Okay. So these cells are present underneath the upper epidermis of plant cells. Okay, now we know that there are many chloroplasts, right? At the ends, we have a large number of chloroplasts. So why do we have a lot of chloroplasts? We have these
Ma'am,
lot of
for
chloroplasts.
maximum,
Yes.
And because so, so that maximum sunlight can be absorbed for
Yes.
photosynthesis.
To absorb maximum light. And as they are going to absorb maximum light, what will happen? There will be more photosynthesis and there will be more food production for the plant. Then we have the ciliated cells. What are these ciliated cells used for? Like, how do they look? First, we'll have a look at how do they look. They're like this. And then you have tiny cilia over here at the top. So here you have the nucleus. First, where they are present, they form the lining of nose and windpipe. Then you have, like, you do have these tiny cilia inside your nose and your windpipe, you can say either it is the respiratory tract, right? So these tiny cilia, they are in continual flicking movement, right? Now, due to that flicking movement, that is going to create a stream of fluid.
It's the
That is
mucus.
the mucus. Very good. That's the mucus. And what does mucus carries? It carries dust and
Plus,
Microbes.
bacteria. Okay. Okay, then due to this dust and bacteria, what happens then? Due to its flicking movement, the cilia will move in a flicking movement, right? And the bacteria and dust particles with the mucus, they will be pushed to the stomach, right? And where we know the stomach contains an acid that is called HCl, right? That is the hydrochloric acid. Hydrochloric acid is present over there to kill the bacteria. So that would be killed inside the stomach. Those bacteria would be killed inside the stomach. Then you have the nerve cells. How do these nerve cells look like? And there's the right here's the cell body. So we call this a cell body. This middle one is the nucleus. So you call it a nerve fiber. So what are nerve cells used for? They are specialized for conducting what? For conducting nerve impulses. The messages, or you can say the electrical impulses, they travel along these nerve cells. And these nerve impulses travel from the brain and spinal cord. So usually we know these fibers, nerve fibers, they are here. They are long, right? They are long. Due to that, they connect distant parts of the body.
Ma'am,
Yes.
ma'am, isn't this the myelin sheath?
Which this nerve fiber, yeah, that's the second name for that. This nerve fiber is also called as the myelin sheath. Now we know there are some chemical reactions happening. So those chemical reactions, they cause the impulses to travel along the fiber. Then we have the sperm cell.
Ma'am, could you go the last point of the nerve cell?
Like
Yes, yes, yes.
the chemical reaction that cause the impulses to travel along the fiber. Okay, for sperm cells, we know its head is in a streamlined shape. Then you have this kind of elongated structure. And at the end, you're going to have a long tail, right? So we call this head as acrosome, right? And this is the mid piece. Inside that mid piece, you have there is the presence of mitochondria. And then at the end, you have the tail,
Ma'am,
right?
Tail.
ma'am, isn't this called the flagellum?
Yes,
Ma'am, isn't the tail called the flagellum?
yeah. No,
Yes.
this tail is actually, I'll tell you that.